Hey guys, it's Mr. Santiago. I just wanted to do a quick lab demonstration video for you guys to show you how scientists can check food to see if there's something like proteins or starch or sugar in there. All right, we're going to focus mostly on starch today just because it's a simple demonstration and it's quick and easy to do. But just really quickly before we do that, I want to just make sure that you understand how these macromolecules are put together. Okay, and so we have carbohydrates, proteins, nucleic acids, and lipids. And so these things are made by individual pieces that are put together. And so when you create something like carbohydrates, for example, if you have a monosaccharide, let me just see if I can use this as an example. Get this all. Here we go. Let's pretend this paperclip is a monosaccharide. Okay, this is just a simple monomer, right? A simple building block, right? It is a carbohydrate itself, right? But it's also used to make larger molecules of carbohydrates like starch and cellulose, right? Starch is the long chain of little sugar molecules, right? That are put together, right? And plants store those for energy, right? And then you have cellulose, which is very similar, long chains of these individual monomers of glucose, specifically glucose sugar, that are made to create this plant cell wall, right? It makes a good strength and structure for trees and wood, all right? And so in any case, so this is how carbohydrate chains are made, like starch and cellulose. You have these little monomers, you put them all together and you make a big chain, right? And these are carbohydrates, also known as sugars, but they're made of very simple sugars, right? Little pieces, right, that are put together, right, to make the longer, bigger macromolecule pieces, okay? And so the starch is actually, so if we pretend that this is starch, that's actually what we're going to test today. We're going to use some iodine to see if, I have some test tubes with some things in there. And we're going to see if, what it looks like when iodine comes in contact with starch. All right. In any case, if you wanted to make, say, like a protein chain, you'd have to use a different monomer, a different building block. You can't use a monosaccharide. Monosaccharides are sugars that are only made, create carbohydrate chains. Let's just pretend that this is an amino acid. You can see quite different than you saw with the monosaccharides here. So an amino acid is what we use to put together, right, by peptide bonds. If you remember, amino acids are connected by peptide bonds, right? And this is what creates all different kinds of proteins. Okay, like enzymes and other types of things. Okay, and then we have nucleic acids, which again, the building block is different. The monomer is different. The monomer for making nucleic acids are nucleotides. So these nucleotides, again, same idea. You put them together, repeating one after the other, right? And you make these long strands of RNA or DNA, right? These are nucleic acids. And then we have fatty acids or lipids, I should say, are a little bit different, right? Lipids have a single glycerol head group. Okay, I'm just going to use this little plastic model piece here to show you, to represent a glycerol head group. But then they also have fatty acid tails attached, right? Some of them might only have one one fatty acid tail. Some might have two, some might have three. But in any case, the fatty acid tail is just really carbon hydrogen all the way down. It's just a chain of carbon hydrogen all the way down. All right. And so that's how lipids are created. So a little bit different than what we saw with the other three macromolecules, but you do need to know all of these. Okay. All right. So what are we going to do today? We're going to go ahead and take 4 test tubes. I already wrote out a diagram on the board. We're going to put, I put water in one of them. I put salt in another, sugar in another. This is just simple sugar. Well, actually, let's go through this. Water, I put distilled water. Okay, distilled water is purified water that has no minerals. Everything's been taken out. Not the kind of water that you would normally drink at home unless you go out and buy distilled water to drink. But in any case, it's just pure water. And the reason why we want to use this, we want to include this, is because we want to see what the iodine looks like when we put it into just pure water. Okay. And do I have enough iodine here? I might have it. I should have enough. All right. And so that's so that we can compare the others, right, to something that we know is not going to have a chemical reaction, right? We know iodine is it's not going to have a chemical reaction with just the water. So we can see what it looks like and then compare the rest to it. All right. So we want to add water as our negative test here. And so then we have salt, which is just something you would buy at the store, right? It's just basically table salt that you would use for cooking, right? Put on your food. So I mix that in some water in a test tube. And then we have some sugar, again, same type you would have at home, granulated white sugar. Some of you have different kinds of sugar, but basically this is just standard white granulated sugar that, again, you would use for cooking or baking or putting on your food or in your coffee. All right, and then we have starch, right? And so we already know the iodine should react with the starch, so we're going to take a look at the difference. We're going to see what it looks like when it does react with the starch. And this is just something, again, you could buy at the grocery store. This is just cornstarch, okay? We took a little pinch of that, put it in a test tube with some water, mix that in, and we'll see what that looks like. All right, so let's go ahead and get started. We're going to start with the water first. Let's get some iodine in a dropper here. All right, so here's just pure water, as you can see here. Put some drops in there. Shake it up a little bit, help it mix a little bit. And you can see it turned, now iodine itself is this color. Here's the pure iodine in my dropper. Let me see if you can see that there. Right, there's a pure iodine, right? And so when you put it in water, right, the water just takes on the color of the pure iodine. So no reaction occurred. It's just diluted iodine in here, right? All right, so we can use that as a comparison. Now let's go ahead and move on to B right here, salt. I mixed in some of that table salt in here. I put a little too much. You can see some of it's at the bottom. That's okay. It doesn't matter. It's not going to affect anything. So let's put some iodine in here. And you can see same basic thing is happening, right? It's just turning yellow, just like it did with the water, right? And you know, if I give it time, it'll kind of diffuse all its way down, right? But you get the idea, right? It's just, you know, turning the same, basically the same as what we saw with the water, right? So not really much of a difference there. All right, then we're going to go with the sugar. And again, same thing, right? You can see if you give it time, we mix it, it'll mix in, but no different, right? Same as the salt and the pure water. Now let's take a look at the cornstarch. Look at the difference there, right? Put some iodine in that, and very drastic difference, right? This turned like this. It looks black, right? It looks very black, but in certain light, it looks almost like a dark bluish black. Okay, but this is the reaction that we have when we have iodine and starch. And so what we can do is, if you wanted to test food at home, for example, you have some bread, maybe, right? You know, chop it up into, or take some breadcrumbs or something, right? Real fine. You could just, or just have a slice of bread. Just if you had some iodine at home, you could, you know, just basically put drops of iodine on it, right, and see what color it turns, right? Some people might put the bread in a blender with a little bit of water and you get like a soupy mixture and then do it, but I mean, there's no need to do that. I mean, if you have cereal at home that has starch in it, like Cheerios, for example, has starch in it, you can basically just put some drops on your Cheerios, right? And then it would turn that dark purplish black color. Okay, so this is one way that scientists can actually test to see if there is starch in foods. Now, there are different chemicals we can use to test for proteins and for simple sugars, right? Now, notice, this is interesting, right? Starch is made of simple sugars, monomers of monosaccharides, right? But yet we put sugar and it didn't react, right? We put one of the test tubes, right, had sugar, test tube C, and it didn't react. And that's because iodine specifically reacts with long chains of glucose. Glucose is the specific type of monomer. Long chains of glucose, right, all bonded together, and that's what it reacts with. It doesn't react with individual, smaller, different types of monosaccharides, because there are different types of monosaccharides. You have glucose and fructose and galactose and maltose and different types of things like that, right? In any case, this is basically it. This is just one little quick demonstration. I just wanted to show you, just to let you know how we can use little lab experiments to determine the content of food items. All right, so thanks for watching, guys. Don't be surprised if you see a quiz question or some question somewhere regarding this demonstration video. So if you didn't take notes on this, you might want to go back and watch it again, maybe take a few notes, okay? And have a good day.